Thank you very much, Marta. Thank you very much today for being here. It's really exciting opportunity actually to be here with you, with this audience. I know you're all hungry, so this is the last talk and we're almost there. So thank you very much for your attention already. Okay, so I'm going to talk about electronic lab notebooks. So I want to first give a very brief introduction about who I am. I obtained my master's degree from Radboud University Nijmegen, where I studied molecular mechanisms of disease. And during this master's program, I performed 2 research internships where I studied virology and molecular immunology, as well as stem cell biology and transcription. Then I moved to Erasmus MC, Erasmus Medical Center, Rotterdam, to do my PhD. And during my PhD, I studied UV light-induced DNA damage and repair. And to address my research questions, I regularly use techniques such as proteomics and microscopy. And as a result, I generated a lot of data myself. At this moment, I work as a data steward at the faculties of Applied Sciences and Mechanical Maritime and Materials Engineering, where a wide range of research topics are covered, such as biotechnology, quantum nanoscience, biomechanical engineering, and cognitive robotics. So when I'm talking to researchers, I always like to first relate to my own experiences of being in the lab. So here you see a photo. This is a photo made from my office during the time I did my PhD, and you see a shelf full of paper lab notebooks. So if you're curious how these paper lab notebooks look like if you open one, well, I'm sorry, but they don't look very different from this example of more than 100 years ago. So what's the problem with paper lab notebooks? So first of all, they are paper, so they are not digital. You can't easily search through them. And they are mostly handwritten, so you can imagine some people have pretty handwriting and some people have ugly handwriting. I do. And also, I have observed it in Holland that some people actually write it in English and some write it in Dutch. So you can imagine that it is not very readable, actually. And also, another important point is that they can store only a a printed copy, printed and actually literally glued on the paper lab notebook of the digital data set. And that's not reusable. So that's why when I talk to researchers, I ask them questions such as, actually, what do you do when people in your research group leave? Are you able to easily find the data that you're interested? And let's say you found the data. Does it mean anything to you? Is it documented in a proper way so that it makes sense? So actually, I'm very, very lucky that today Danny explained this reproducibility crisis issue in a very nice and comprehensive way. Yeah, so it's according to this survey, actually, more than 70% of the researchers have tried and failed to reproduce another scientist's results, and more than 50% have failed to reproduce their own experiments. So what factors actually contribute to this irreproducible research? And they were also very nicely portrayed already by Danny, such as the selective reporting, pressure to publish. But today I'm going to more talk about methods and codes being unavailable, as well as the raw data not being available. So I think it's important here if we look at how the research life cycle goes. So this is an example of how my research data look like during my PhD. So on the left-hand side, you see a microscopy image of cells. I hope you can see it. It's a little bit bright here. But anyway, so that's my raw data that I make an image of the cells, and the cells are red. They are red because they have a fluorescent signal. So normally what I would do is I would quantify that fluorescent signal and end up in an intermediate data table with numerical values. And from there, I would generate a graph where I compare different conditions. As an example, here it is shown as ABCD. So what happens is this. Most of the time, researchers only publish this final data graph. That's not reusable. So therefore, I ask researchers, actually, the data you publish in your papers, is it reusable? And if not, are you able to go back and find the previous data stages which are reusable for other researchers? And maybe let's not think yet about all being so open now for the whole world. Let's say the person who generated the data in your group has left, and now you have a new lab member, and that new lab member wants to work on the same data. Are you already able to find it for that purpose? What about the generated samples? During research, researchers generate all sorts of samples. It could be cell lines, bacteria, protein, DNA samples, synthesized chemicals. So you can imagine that the person who generated those samples is gone, and you have a new lab member. That lab member needs to work with such samples. Do you have a proper system, digital system in place that persons can easily search through and find them? I mean, they're not going to go through all of the freezers to find the samples that they're interested in, right? And what about experimental and measurement parameters? So normally when researchers publish a paper, in the materials and methods section, they need to explain how they have performed their experiments. Is that information enough for other researchers worldwide to reproduce your results? If not, or is it enough actually already for you to reproduce your own results? And let's say if it is not enough, do you at least have in your own lab environment a good documentation system explaining so that you can reproduce your own results? Well, actually, there are digital tools available that helps researchers to tackle all of these issues. They are known as digital notebooks, also electronic lab notebooks. And these are digital, therefore they are searchable, they are readable, they are organized and structured, and they are traceable. So it means that every single change that's made can be traced back. And therefore it is fraud-proof, and therefore it has version control. And it's possible to link all of the data stages with each other. It is possible to link the data with the samples. It's possible to have metadata recording, as well as team collaboration tools and mobile applications. This is why on 15th of March we organized an event dedicated for this electronic lab notebooks. You can find all of the material available if you're interested, all the presentations, and I have written a report about this event and you can read the report as well. So we were very happy actually that the event attracted a lot of attention. Just like today, the tickets are sold out. And what was even nicer to see that after the event we got contacted by researchers from various Delft departments because they asked us how we can implement in our daily practice using these electronic lab notebooks. So this is exactly the cultural change we are talking about, right? So that's why at this moment we are in contact with the researchers to be able to determine what they expect and require from an electronic lab notebook. And our next plan is to start a pilot study accordingly. Sorry, so no questions yet, I guess, but this is all my information and I would be very happy to answer any of your questions. You can contact me, you can read the reports, and that's it. Thank you very much.